Thank you, Deb. That was beautiful. And finishing up now on the scientific legacy of Theo's work, Chris Kassotis, who's worked with Susan Nagel and really dug into this question of endocrine disruptors and fracking fluids and pulling together what Kim started us out with, this fossil fuel connection, the fact that so many of our fossil fuels are also endocrine disruptors, and that we really need to be thinking about these industries together and tackling these problems as part of one and the same issue. So Chris, please join us. Thank you.
Well, I have to thank the organizers for having me out here. It's been a very interesting morning so far, and I look forward to hearing from everyone in the next couple days. So as Sara said, this will be a little bit different in that I'm kind of on the lab side of things and have had limited interaction with kind of the communities that are most affected. But I'll touch on some of that, and I'd be happy to talk about that more over the course of the next few days. So really, I'm just going to briefly kind of introduce the idea of endocrine disrupting chemicals as they relate to oil and gas development, and then kind of touch on the three branches of my work. So chemicals used in the process that can act as endocrine disruptors, what that means for water quality in these areas, and then lastly, looking at some actual health effects in a mouse model. So I kind of wanted to briefly talk about, how I ended up here. As others have said, this entire field of endocrine disruption research started back in 1991 with Theo Colburn. So this meeting of scientists was spurred by her, and ultimately came to this, the wing spread statement that kind of began this field. So I just have this quote: "A large number of man-made chemicals that have been released into the environment, as well as a few natural ones, have the potential to disrupt the endocrine system of animals, including humans." So again, Theo really spurred this, got together a collection of scientists and key people to push this field out and get it going. One of those being Fred von Saal, who's pictured here on the right, and I'll come back to him in a moment. I started my journey here in about an hour and a half, 2 hours north of here. So I did my bachelor's in Keene State College in southern New Hampshire. And Susan Whittemore there, my professor, has done and continues to do endocrine disruption research in frogs. But she got me really interested in this during my college time, specifically with conversations on endocrine disruptors. And I'll always remember Lake Apopka, which is kind of a very famous example of endocrine disruption, in that case of alligators exposed to industrial chemicals, spearheaded by Lou Gillette's lab. And ultimately, then I did some small work on Siamese fighting fish exposed to diethylstilbestrol, which is a very potent synthetic estrogen, and looked at their changes in behavior as a result of the exposure. But ultimately, that just led me to come out to University of Missouri, which is where Fred von Saal is. And interestingly enough, he then trained Susan Nagel as a PhD student, 20 years ago. So he's still there, and I've had the pleasure of having both of them, kind of two generations of researchers, as my co-advisors, and came out with an interest in looking at mixtures of endocrine disrupting chemicals, and led ultimately to me working on this hydraulic fracturing as a kind of a great example, great but not so great example of an environmental mixture of these chemicals. So just to try and touch on a little of the basics here. So hormone action is of course very complicated, but if you think of some of the common receptor systems, so the estrogens and the androgens people are most commonly familiar with, I think, in terms of the male and female sex steroids. But glucocorticoid receptor, progesterone, thyroid, they're all very interconnected, and they all are necessary for normal development and normal physiological processes throughout life. So the Endocrine Society defines then an endocrine disruptor as a chemical or mixture of chemicals in the environment that interferes with any aspect of hormone action. And it's an important distinction because previous definitions all include that there is a requirement of some sort of adverse health effect to actually say that something is an endocrine disruptor. The Endocrine Society then has taken that out and says, well, any perturbation of the endocrine system is adverse. You don't necessarily need to know, what that adverse health outcome is to define an endocrine disruptor. But this can happen through two different main mechanisms. So the first is direct interactions. So hormones act by binding to a receptor, and that receptor binds to then a response element on DNA and results in some biological effect. So these chemicals can act as receptor agonists. They can bind to a receptor and drive that normal biological response. They can also act as antagonists. So in this case, they can prevent the normal binding of a hormone to a receptor and prevent then that normal response. And then the other real mechanism is through these indirect interactions, and these are they're varied. So these chemicals can influence the expression of the receptor in specific tissues, hormone levels throughout the body, and even the response of the receptor to binding the normal hormone. And all of these things can lead to then a number of different adverse health outcomes, right? And everything from cancer incidence to infertility, subfertility, immune disorders, metabolic syndrome has gotten a lot of attention in the last few years. So that's obesity, cardiovascular disease, and diabetes. And there is good evidence then for mechanistically why specific chemicals can lead to these adverse health outcomes. And at the same time, we see then growing trends in many of these health outcomes in humans. So there's been a lot of interest in exploring this relationship between exposure to these chemicals and the development of these outcomes. It's important to say then that a lot of times in the lab, we focus on receptor binding. So that's the simplest thing for us to look at in the lab. However, it doesn't tell the whole story. So it often takes going to these whole organism models to really get at these adverse health effects. So we started this really largely because Theo called up Susan and said, you need to be working on this. So we've always had a good relationship with Theo, and I've had the pleasure of working with her now for a number of years. But it was really this first study, and we had gotten information from her before this point that highlighted that, of the 353 chemicals it looked at in this study, 37% of them were known or suspected endocrine disruptors. So there was some evidence that they could influence the endocrine system. So we set out to do a couple things. One, we set out to see exactly how those chemicals interacted with the endocrine system. Whether those chemicals, if they did interact with the endocrine system, could influence water quality in those areas. What I won't talk about, but is certainly ongoing, is measuring and assessing chemicals and concentrations of chemicals in both wastewater and drinking water, and then looking at potential adverse health effects for humans and the animals living near these operations. So a lot of the work that we do, as I said, is receptor binding. So in our system, then, we're using human breast cancer cells, and we use a chemical reagent then to insert pieces of DNA into the cell. So one of those is a human hormone receptor. So for this example, we'll use the estrogen receptor. And the other then is a human response element. So then the estrogen response element tied to the luciferase gene. So the basic interaction here, if we follow, shown here in the little circle, is 17-beta estradiol, kind of the natural estrogen in our bodies, binds to the estrogen receptor. The receptor then translocates to the nucleus, recruits coactivators, dimerizes, and binds to, ultimately, this estrogen response element on DNA, and drives the production then of estrogen-regulated genes. In this case, we have it tied to the luciferase gene. So hormone binding then results in the production of luciferase, which we can then measure in the lab as luminescence. So we can then get a luminescence value for a chemical or water or anything that we're interested in looking at, and correlate that back to the amount of hormone that bound. So just to kind of show you that graphically, if you look at the solid line, we've got 17-beta estradiol, which is our natural estrogen agonist. And you'll see then with higher concentrations you get complete full production of luminescence, 100% luminescence at the highest concentration, and then progressively less at lower concentrations. So we can construct this curve of a dose-response curve, so to say, that will let us compare other chemicals or water samples to. We can then also do that in reverse. So we take then the half maximal concentration and we set that equal to 100%. We add it into all of our samples, and then we'll add test chemicals or water samples to that. And then if you look at the dotted line, we then measure inhibition of that luminescence. So we know what luminescence is produced by this concentration of our estradiol, and we can then measure an inhibition of that signal in the lab. And this is just to say that now we do that for 5 receptors. So estrogen, androgen, progesterone, glucocorticoid, and thyroid. So just to touch on the 3 branches of work that we've done. So we started with hydraulic fracturing chemicals. So we selected then 24 chemicals with TEDx commonly used in the fracking process, and that had some suspicion that they could be endocrine disruptors. And ultimately found then that we had very little chemicals that were able to bind to and activate receptors. So 1 for the estrogen receptor, 1 for progesterone, and 2 for thyroid. Most of the activity that we saw was antagonism. So these were chemicals preventing the normal hormone binding to receptors. So we saw this then for 23 of the 24 chemicals that we tested, and importantly we found that these activities occurred at levels actually found in the environment. We also then looked at water, seeing could we detect those signatures of endocrine disruptors in water. So in this case then we collected 1 liter water samples, filtered them, concentrated them, and applied those water samples then in that same assay that I previously described. So we started then in Garfield County, Colorado, really with the help of Theo. So for people that work in a lab, forming those personal connections on the ground is really challenging and takes a lot of time. So having people that can help with that process can really make all the difference in the world in terms of a successful sampling effort. So we set out then to examine then densely drilled sites in Garfield County that had also experienced a leak of some kind of hydraulic fracturing fluids, and then also collected some reference sites immediately outside that area, and then also from Missouri. And ultimately, at this point then we were limited to estrogen and androgen receptor activities, but ultimately we saw then very little activity of any sort in the Missouri water that we collected. This is groundwater. As we moved into Colorado, we saw a significant increase in that estrogenic activity or activation of the estrogen receptor. But as soon as we moved into that box, that dense drilling spill site, we saw essentially saturating levels of estrogenic activity, so very, very active estrogenic activity, and then the introduction of this anti-androgenic chemicals that blocked the normal action of the androgens or testosterone. Surface water was a very similar story, except within that drilling dense region, when we also see then the anti-androgenic activity, we see the introduction of anti-estrogenic activity, which again we see very little of in our reference sites. So we've done a lot of other sampling in other regions that I'm happy to talk about over the course of the next day or so. I'll suffice to say that we've done a much more comprehensive sampling effort now in Colorado. So we have about 60 unique samples now, 75 sites, and I've just finished up analyzing that, and I'm putting the data together. So should have something more to share soon. So again, we're seeing elevated antagonist activity, so chemicals that block receptor activity in surface and groundwater in regions with oil and gas wastewater influence. The last little bit that I'll just mention briefly is this developmental exposure. So we wanted to get at, well, what does it actually look like in terms of adverse health effects for an animal exposed to a mixture of these chemicals? So we exposed then pregnant mice starting on the 11th day of gestation, birth is gestational day 18, to a mixture of then 23 of the 24 chemicals that we have evaluated in the lab via their drinking water. And then following birth, assessed offspring then at a number of different endpoints. I don't have time to go into all of them. We'll just highlight some of the changes that we're seeing in male mice. So we have increased body weights in mice exposed to this mixture. After controlling for body weight, we still have increases in heart weights, thymus weights, and testis weights. And that is also correlated with a decreased sperm count in those mice, as well as we see a slight acceleration of puberty and decreased anogenital distance, which is a marker for anti-androgenic exposure. So as overall take-homes, we've shown then that some chemicals can activate or inhibit normal hormone action. These chemicals may increase receptor activities in these drilling-dense regions, and that gestational exposure to a mixture of these chemicals, importantly at likely environmentally relevant concentrations, resulted in adverse health outcomes in male and female mice. So I'll end just by telling you, this all started back with Theo at the Endocrine Disruption Exchange, getting us interested in this. But it's really expanded, and there are other groups around the country that are similarly expanding. But I just want to highlight some of where our research has gone. So as I said, we've done a lot of work in Garfield County, Colorado, so we've formed now a lot of collaborations with landowners, citizens groups, and even high schools in the area. We're working with Deb Thomas in Pavilion and Wyoming to look at water quality there. We've partnered with the USGS headquarters to look at water quality in West Virginia and North Dakota. Charlotte Patton has been great in helping organize a lot of these sampling efforts, and we've been talking to her about doing some sampling now in California. On the lab side of things, we have a collaborator looking at the glucocorticoid chemicals that we have identified and metabolism or development of obesity. We have a lab at University of Rochester looking at immune function. At UMass Amherst, a little closer, we have people looking at thyroid inhibition, as well as the breast cancer development. At Duke, we have Avner Vengosh, who's been quite well recognized for his work showing then that his ability to show that contamination is specifically due to fracking and not more conventional oil and gas production techniques. And then Dr. Heather Pattersall at North Carolina State on brain development. And then a lot of collaborators locally at the University of Missouri. So we've formed a collaboration with a extremely talented chemist who has been working on development of analytical methods to measure these chemicals, non-targeted methods to identify new chemicals that haven't previously been reported, and also developing methods to look at biomonitoring in humans, so blood and urine, which, as we heard, is really needed to determine exposure. And with that, I will end and just thank the EPA STAR predoctoral fellowship that I got has really let me do this work. Although it should be noted that they now have a special exemption that no one on an EPA STAR fellowship can now work on fracking. Yeah, don't know what to say about that, but I will end there.
Thank you so much. Do you have time for questions? Okay, so we have just a few minutes for questions. I'd like to invite the panelists to just come up the front here. And we've got a couple of circulating microphones. We'll keep questions to 10 minutes. So thank you, Chris. I can't imagine a better way to end this than really thinking about how this is informing scientific progress. And just on other sad notes, citizen science has now been banned in Wyoming. So there's real need for active resistance. Do we have any questions? We've got one back there. Boris?
Hello, everyone. My name is Elizabeth Wilder. I'm a second-year PhD student here at Northeastern and part of CIRI. And I want to say thank you for all of those wonderful presentations. The question that I had was, we've heard presentations from some social scientists, we've heard from biological scientists, we've heard from citizen groups and organizations, and one of the things that seems clear is that in this case, how central Theo was to these kinds of collaborations in pulling together people from all of these different disciplines and in working with communities. And I was wondering if perhaps the panelists and maybe even some of the audience members who we have a range of expertise in this room could reflect on some of the principles of pulling together those collaborations. So how can we put into practice some of the things that Theo did? And one thing that I thought of, something that Deb said, sort of begins with the community are the experts on their experience. So that's one thing that I definitely took from it, from all of your presentations. But I'm hoping maybe we could hear a little bit more about what others think.
Great question. Kim, you had such direct experience with Theo. Did you see her do any practices in the community organizing?
Oh, well, one thing Theo had was years of experience working with scientists. And so she knew people all over the country and even a lot of people in Canada. And so she already had this wonderful network of people that when she wanted someone to do specific research, she could just talk to them and get them motivated. She was such a motivating person. And so that's good. And a lot of people directly approach us, and they still do all the time. And we try to be as helpful as possible and just work with as many people as possible who want to be involved with us. Everybody. Yeah.
Deb, do you have anything on that?
Well, I just think she was, and I touched on this, I mean, she was just so amazing because she would look at anything you had. And I think that it's really easy when you're a professional person to discount things, maybe. I'm not a scientist, I'm not a professional anything. So, but I know that many times it's hard to get scientists or academics, or especially health professionals, to really consider what you're seeing on the ground. And she was not there. I mean, she was like, yeah, bring it on. And then she would like analyze it and make you do your homework and put together these just amazing pieces of data and information that really showed you what the picture was. She was truly amazing in that aspect.
Yeah, I don't think it's incidental that we saw, I think, 3 or 4 times the bar charts that TEDx put together, and part of what they did was report data in a very clear way that was easy to interpret and easy to read. And so I think they put a lot of time and effort into thinking about how to demonstrate their concepts. So using a pie chart that shows, okay, only 1% of this large pie do we actually know the whole chemicals around. It really helped to kind of express the information visually. Chris, do you have anything there?
Yeah, and I think one of the things that we've talked about, or several of the speakers have talked about, is then bringing this back to the public, right? So ultimately, we've done a lot of work in Garfield County, Colorado, and as I kind of alluded to in my presentation, making the for people in the lab, making those connections to people that are living in these areas is really difficult, right? And in fact, certainly some of them have had so many people come up to them and say, we want to test for this, that they're almost to the point of hostility in some cases. They do not want you on their land. They do not want anything to do with you taking their water. So bridging that gap between the two is something that I think Theo is really good at. Convincing people this is a good idea, this needs to happen. More information is always better than less information. As I think Sara alluded to, that there's this big disconnect, right, between the paper that I will publish in a scientific journal and what it means for people living in those areas. So we've made a point then of spending a considerable amount of time generating, essentially heat maps of, what we see in the water from people's households and what it might actually mean for their health. And from our original study anyway, it was really well received. Everyone seemed to make sense of it and welcome us in when we went back in 2014 and help us make new connections and sample new houses. So I think it's really, in our case, about bridging that gap between the scientists and the people living in those areas.
Great. Another question here with Sara. You need a drone.
It's not a question so much as a comment. First of all, really appreciated the range of topics that were covered in this panel. We've had Deb and Sara at NIEHS, and Kim, thank you very much. But I particularly wanted to call out Chris for making very technical information accessible. It's clear you've been talking to the public, and you really explained things very well in a way that made the connection between what people are experiencing and the science. And it was particularly heartening to see your final slide about the collaborations and how this consciousness has grown for you about the need to connect with the people that your research is ultimately going to benefit. And I really appreciate the range of information that you provided in this panel. Thank you.
Thank you, Sara.
Thank you.
Thank you. In the middle there.
Hi, my name is Anna. I just want to thank all of you so much for being here today. This is amazing. I feel like I found my people, which is why I had to come from Northern Ontario, Canada to find all of you. But I have founded my own organization called Gumption, and we're doing something very similar to you. We're creating a mobile app right now that's trying to integrate local stories to complement natural scientific data and knowledge. Perry Sound in Northern Ontario is a very sort of unique dynamic in a community, whereas with a population of 6,200 people, there's an old munitions factory there that they had been dumping a lot of chemicals and everything into the environment for over the course of 75 years, but that ended in 1985, so not many people know about it anymore. There's also, it's heavily cottage, so there's a lot of high-income residents, low-income residents, natives and non-natives. We're now starting to engage the local community and educate them about that, the local stories. Everyone seems to know that there's a health issue and that there used to be this old property, but they haven't made the connection between the two. So now that we're starting to, ask questions and make connections, people are starting to become very fearful of what that means for their real estate value. So I was just curious, particularly for the low-income residences where their entire life savings is their home.
Yeah.
So I was just wondering if you could give me any guidance on how to handle that in that community engagement aspect.
Yeah.
Thank you.
Yeah. Deb, I wonder if you have any input there.
That's a really tough one, and you nailed it, because many times people have everything that they worked for their entire lives wrapped up in that community. In many of the places where I work, they also have this incredible sense of responsibility to place, because a lot of these people have been on the land, and their families have been on the land for many, many years. And so, you can't force it. We do a lot of the work that we've done in terms of health surveys and even testing, we do without taking names. We tell them, you know, if you want to just give us your information, we won't share who you are or what's happening with you, because it's really important. That anonymity is really important to them, and I think the more they find out, the more secure they become with it all, the more they want to speak out. Particularly the people who have that strong sense of place. A lot of people just come into it, buy it, you know, don't have that. And that's why in some places where, like the Bakken boom, where people are making lots and lots and lots of money, it's really difficult because everybody's on a 5-year plan to make $1 million, you know, and then they're gone.
Yeah. And I would say that if you're making a digital platform, you know, there's lots of ways in which, when we designed WellWatch, we also thought about anonymity. So not requiring email addresses, not taking IP addresses, not storing IP addresses. If you're making a report online, fuzzifying the location, so saying you could just make a report in a county, not for a specific location. Giving people the ability to change their date range on complaints, because dates can help you. So I think there's a lot of technical approaches you can take to protecting anonymity, and in fact Phil has an entire study on that, so you should chat with him about data privacy and anonymity in online settings.
Can I just follow up with that? Another really difficult thing in this work is it's really easy to make it about you instead of about what you really should be doing, which is helping these people who are so impacted. And so I think it really helps if you get centered back to making it about the people that are living with this or who are the most impacted, rather than, you know, your work or your research or whatever.
Yeah. So we're out of time. I see one desperate question in the back, so we'll take one last question, and then I would like to propose that we have a breakout session for anybody who's interested in talking more about fracking and health effects. Claire, go ahead.
Hi everybody. My name is Claire Miller. I'm a community organizer here in New England.
There we go.
I spend a lot of time right now working with communities that are facing fracked gas pipelines, fracked gas compressor stations, fracked gas power plant proposals, and it's been incredibly inspiring to hear about this collaboration, this work. I've already grabbed a couple of the DVDs to share with folks later tonight that I'm going to see, and just incredible. So my question is, I guess, for the whole room, which is, if you're looking to try and get more connected to these communities and be part of these collaborations, please consider joining the HERE database, which is the Health and Environment Assistance Resource Database, because I'm not a scientist, I don't have technical expertise, but I want to connect folks like you with the folks who need it most. So there's handout is over on the table over there. Come find me. Thank you so much.
Thank you.
Spoken like a true organizer.
Thank you, everyone. Thank you so much for coming.